Hepatitis. What is hepatitis? Hepatitis is the inflammation of the liver. The 6 different known strains of hepatitis are hepatitis A through F. Hepatitis C is the fastest growing concern among North Americans. For this presentation, we will be focusing on hepatitis C. Normal physiology. The liver is formed of units called hepatic globules, which consist of single liver cells called hepatocytes. Liver cells filter bacteria and toxins to make them safe. They are used to get nutrient-rich blood into the hepatic globules to extract nutrients from metabolism or storage of macromolecules.
Progression of hepatitis. HCV enters through the bloodstream and disrupts homeostasis in the liver. In the acute phase, the virus is asymptomatic. If not eliminated, the virus progresses to chronic hepatitis, leading to the destruction of the liver. How does this occur? Risk factors for hepatitis include contaminated blood transfusion from the 1980s, sharing drug paraphernalia, improper sterilized needles for tattoos or piercings, or ingesting contaminated foods and beverages.
Let's take a closer look. Pathophysiology. Stellate cells are antigen-presenting B cells that stimulate CD4, helper T cells, and cytotoxic T cells. They present MHCs on the surface to be recognized by T cell receptors. Stellate cells are normally inactive unless the liver is damaged, and then they stimulate proliferation of natural killer T cells. Kupffer cells are specialized macrophages in the liver that break hemoglobin into iron and bilirubin.
Cell cycle regulation, regeneration in the normal liver. Cell cycle regulation occurs when the liver is exposed to external damage and triggers regeneration, which causes little to no scarring. The damaged or excess cells are expelled as waste. Chronically damaged liver. The HCV virus has a higher rate of cycles for regeneration, which lead to collagen accumulation called liver fibrosis. It is the buildup of scar tissue on the liver. Genome instability and cell cycle regulation is disrupted, creating an imbalance in cell reproduction and apoptosis. Stellate cells are activated. They contribute to the inability to remove dead cells, causing scarring that progresses to cirrhosis and creates lesions on the liver.
Invasion of cell membrane. Invasion of cell membrane. HCV virus approaches the cell membrane of hepatocytes. Its outer membrane consists of envelope glycoproteins which attach to the receptors of the host liver cell. The viral lipid coat is fused with the plasma membrane of the host cell. After fusion, the viral protein core begins to uncoat, a process where protein coat dissolves and then releases the viral RNA inside the cell. Positive RNA strands are released. Translation and polyprotein processing now occur. Replicating RNA for viral replication complex occurs in the rough endoplasmic reticulum. New viral RNA creates viron assembly after undergoing RNA synthesis. New HCV viruses mature in the Golgi apparatus. It is expelled through the exocytosis from host cell.
Symptoms. Jaundice or yellowing of the skin and eyes occurs when the destruction of red blood cells and elimination of bilirubin does not occur. Normally, old red blood cells are destroyed and hemoglobin is released into the blood. The disruption of normal liver function creates a buildup of bilirubin, a bile pigment that originates from red blood cells which causes jaundice. Stool is often pale because there is no bilirubin reaching the gastrointestinal tract. It is bilirubin that gives fecal matter the color brown. Dark urine results due to excess bilirubin in the blood being filtered and excreted from the body. Why doesn't the body fight the invasion? Why doesn't the body fight the invasion of HCV? Replication of HCV is very rapid. With a high mutation rate, the virus evades the natural immune defense mechanisms from the host. Due to the mutations that occur during RNA synthesis, the liver cell views the genetically altered viral RNA as its own and does not initiate an immune response to the virus. The viral RNA leaves the host cell and continues to infect more cells. Key concepts. The cell membrane The HCV virus is fused to the host plasma membrane, thereby allowing it to enter the host cell. Once RNA has replicated, it exits through exocytosis. Proteins. Proteins are essential for virus replication of RNA strands, giving the virus the ability to spread throughout the bloodstream. Polyproteins are a key component for a successful viral RNA replication. Bilirubin is a protein derived from hemoglobin that causes the symptoms of Hep C. Homeostasis is disrupted by the irregular cell cycle regulation and inflammation of the liver. It does not easily recognize the viral RNA as it masks a similar structure to those of the host organism. To summarize, hepatitis is transmitted through contaminated needles and food products. Hepatitis in an individual increases the risk of liver damage at a cellular level, and new treatments have begun to emerge. However, there is much information that professionals still lack today to completely cure hepatitis.